So you may have seen a graph like this and may have wondered how to interpret it. In today's video, I will explain what is flow cytometry and for you to understand how to interpret this graph. So first of all, what is flow cytometry? Put simply, is a method of using a laser to count cells. You may have seen a machine like this in the lab. Flow cytometry involves using fluid to pass cells through a tube one at a time and measure the characteristics using a laser and a detector. Often, we will have a heterogeneous population of cells and be interested in characterizing a certain group within that population. As an example, in this case, let's say we are interested in these purple cells. And let's say that they are lymphocytes. We have ones that express membrane protein A, some that express membrane protein B, some that express both A and B, or neither. A common set of objectives in flow cytometry may look like this. 1. To identify and filter lymphocytes, in our case, purple cells. And 2. Of those lymphocytes, determine the proportion of cells that are double negative, A positive, B positive, or positive for both. Before we run our samples into the flow cytometer, it is common to use fluorescent antibodies to label specific proteins of interest. Let's use blue fluorescent antibody for protein A and red fluorescent antibody for protein B. And now our samples are ready to run through the flow cytometer. Our first goal is to identify our lymphocytes. So a common method is to use a forward scatter and side scatter to identify our cell population of interest. Forward scatter detects light diffraction around the cell. Forward scatter intensity is proportional to the diameter of the cell. For side scatter, detectors measure light that is reflected from intracellular structures. Light that is reflected at greater angles, in other words, side scatter intensity, is proportional to the granularity and complexity of intracellular structures. So an example of a cell with high complexity are granulocytes. Let's say we make a dot plot where each dot represents a single cell, and we put on the x-axis forward scatter for size, and on the y-axis side scatter for complexity. We will see clusters for groups of cells, as groups will appear as they have similar characteristics, with larger complex granulocytes on the top right and smaller lymphocytes on the bottom left. This method is called gating. Some researchers will use several gates in order to filter out and identify their population of interest. The one I've shown here is one of the most common methods, but there are many different gating methods that people can add on one after another in order to selectively filter out your population of interest. So now that we have identified our lymphocytes, let's move on to step 2, identifying the portion of cells that express protein A or protein B. As our cells pass through the tube, not only are the parameters such as forward scatter and side scatter measured simultaneously, but also fluorescence is detected. In this case, we have fluorescent antibodies labeling our lymphocytes. If we visualize our 4 categories in a 2x2 matrix by applying again a dot for each individual labeled cell into each quadrant, we will have lymphocytes that express neither protein A or B into the bottom left, lymphocytes that express protein A on the bottom right, those that express protein B on the top left, and those that express both on the top right. And we will end up with something like this. Look familiar? If we come back to our chart at the beginning of the video, this is exactly what it represents, with each quadrant, in this case, on the x-axis, CD3, and the y-axis, CD19. We can see the proportion of double negative cells is around 12%, CD3 positive, 72, CD19 positive at 0.6, and double positive at 14.4. And that is the basic gist of flow cytometry. If you found this video informative and helpful, please subscribe so I know that content like this is valuable to you and will encourage me to create more videos like this in the future. If you have any questions or comments, feel free to post, or if you have any topics you would like to see in a future video, please let me know as well. Thank you.